Hi everyone. We are doing our project presentation, and our project was is called Blue Monday. The project name changed a couple of times, but we went with Blue Monday in the end. And this is because the project is blue carbon mapping of the Niger Delta across the years.
Hi there. Okay, so a quick introduction. Mangroves are critically important but difficult to monitor at scale. I think that this is a topic that a lot of people in well, pretty much most people in our group didn't really know a ton about. So we started out with these three initial questions, which were super vague. How can PACE help to improve mangrove mapping? Can hyperspectral data distinguish mangroves from other coastal organisms? And can we use PACE data to determine if and how mangroves are affected by salinity and temperature events? Next slide, please. Perfect. So the study area that we landed on was the Niger Delta, which is on the west coast of Africa. It is the third largest mangrove forest in the world, and it is dominated. I think I saw that it's 90% the single species Rhizophora racemosa. And we also, like I mentioned, I think we didn't really know a ton about this region, but once we started really digging into the literature and understanding what this workshop is about, um, one of the other things that we found was that it is very, very disproportionately affected by oil spills. We found a data set that showed like hundreds of kind of micro oil spills per year, so pretty wild and extraordinary. If you want to go to the next slide, please. So once we started really figuring out what this was all about and understanding it and going through the literature, we ended up with three very, very different questions, which were much more specific and much more like geared towards the region. And those are, what are the spectral characteristics of that single species, or racemosa? How do oil spills affect the mangrove systems in the Niger Delta? And how does this mangrove system change seasonally? Next slide, please. And I think this is Shulhan's slide.
Correct. So first, we are doing some spectral analysis on the website. We are using a 2020 dataset of mangrove extent. And from that extent, then we are creating 100 random points across the mangrove forest to extract the uh, value of the remote sensing surface reflectance. Next. And on the left is the value of the individual spectrum of each of the 100 randomly chosen pixels. And we average all of that wavelengths. So we get an average mangrove spectrum. Next. And from literature review, we are getting that red edge is the steep slope between 700 to 800 nanometer, which indicates the transition between chlorophyll absorption and leaf internal structural scattering. It is a distinct characteristic of mangrove, so we can use this range of wavelengths to best visualize the mangroves. Next. And we are also doing some classification of the land cover. Here we found that 5 classes is the most optimal compared to the larger classes, such as 10 classes, which on the next slide. We can see that there are more similar spectral signature on the right side, on the right chart. We see many of similar that can be simplified. So I think that 5 classes is the best to represent the land cover from the surface reflectance. Next. And here it is the visualization of the single spectrum on the left, single wavelength, and the composite of 3 wavelengths on the right. We can use this to compare what best to use for the mangrove mapping.
What we started off with, one of the things we did was we created a time series of the spatial analysis. So here we've got the January 2025 to November 2025. And this is just to show the NDVI value. Do you know where the other ones? There it is. Sorry, there's the 2024 one. So this is the entire span of the data that was available from the LandV data of level 3 from PACE. So this is from March 2024 to December 2024. And then you've got January 2025 to November 2025. And I have on the next slide them side by side. So this is the time series of the entire 2024 and of the 2025 data. And you can see here that there is a clear difference across the rainy season and the dry season in Nigeria. So the rainy season is from April to September, and the dry season is from October to March. Now, what you'll also notice, and Ryan, we figured out what it was. In August, there is an August break, and this means so it doesn't rain as much as it would typically in the rest of the rainy season. Hence, why there's a lower index on the 2 August months in the middle of the rainy season. So something we then also started to look further into were oil spills. So here we've got the amount of oil spilled from the start of the PACE mission to present. And this is the monthly oil spill volume. We also had oil spills cropping up in lots of different locations, and you can see it there by volume and location. And what something we found out is that mangrove health fluctuates with oil spills. So oil actually has a really detrimental effect towards chlorophyll. So that's something we would like to investigate a bit more further. So in 2024, from March to December, these are the spills that occurred in the study area. So 23.5% of these spills directly occurred in the mangrove populated areas, and 4.2% of them occurred below less than 5 kilometers outside of the mangrove areas, and then 72.5% occurred past 5 kilometers of the mangrove areas. And then in 2025, there was an average of 37.7 spills a month, with 33.7% of the spills occurring in mangrove areas, 4.6% less than 5 kilometers, and then over 5 kilometers was 61.7%. There was also an average in 2024 of 96.8 spills a month. So, Michelle, it's you.
So, we would like to, because of the short time, we would like to look at other areas concerning this project. First of all, we would like to apply other moisture-sensitive vegetation indices instead of only the NDVI. We would like to apply the normalized difference moisture index, that's your NDMI. We also want to look at the MNDI, that is your modified normalized difference index, to improve the detection of mangrove mapping within the study area. We also want to investigate other ecological stressors apart from oil spill, using other PACE water quality products such as your chlorophyll A, your TSM, and then your CDOM. We also like to integrate the vegetation index with the ocean color and derive environmental variables to also evaluate mangrove stressors within the system, so that we'll know which mangroves are healthy, which ones are under stress, so that we can better do our ecological assessment of mangroves within that area. And then lastly, we'd like to compare PACE with other we would like to compare PACE for large-scale mangrove mapping against other imageries like your satellite, like your Landsat, and then your Sentinel-2, so that we can compare whether PACE can also do a better work than other such as your Landsat and such as your Sentinel-2. So those are those future areas that we're looking at to work on.
And this was just our initial kind of project ideas we had. So we had a sort of range of things going on, and we had possible research areas, and we did end up landing on Nigeria. So what we've learned how to work across different time zones, which sounds super obvious, but it can be really tough, especially like Shulhan, props to you for the fact that I think it's like 20 to 3 in the morning right now for you. We learned how to map and DVI and other variables from place data, how to set up said GitHub repo and Jupyter Notebook, navigating, pushing issues in the process, how to use level 2 and 3 hyperspectral data. And also, we learned that Eli, Ryan, and the whole HackWeek team are absolutely amazing, and a big thank you to them. But that is everything as our bibliography. And just for Ryan, here is the last slide.